Chapter 10, Segment 1 begins with discussing the vertebrate eye acts in some ways like a camera. The eye having both optical functions and neural functions. First structure addressed is the cornea, which performs refraction, which is the bending of light rays by a change in the density of a medium, such as the cornea and the lens of the eye. The lens is the second structure, and it helps focus an image on the retina. Ciliary muscles are the muscles that are used to change the shape of the lens, which is called accommodation. When mammals age, their lenses become less elastic, so then they're less able to change the curvature to bring nearby objects into focus. The pupil is basically the hole that allows light to enter the eye, and you can control the amount of light that enters the eye within the size of the pupil. The iris is what surrounds the pupil and allows it to open and close. The belladonna is a drug that blocks acetylcholine transmission within parasympathetic synapses of the iris and relaxes sphincter muscle fibers and permits the pupil to open widely. Extraocular muscles are one of the muscles that are attached to the eyeball, and it controls the position and movements. The next section is visual processing, begins in the retina. The retina is where the first stages of visual information processing occur, and it contains photoreceptors and neurons. A photoreceptor is a neural cell within the retina that responds to light. This can either be cones, rods or cones working best at low levels of light, and cones providing us with color vision. Both release neurotransmitter molecules to control activity of bipolar cells that synapse with them. These bipolar cells are interneurons, and they pass information to ganglion cells. And ganglion cells, their axons form the optic nerve, and the optic nerve extends from the retina to the optic chiasm, which provides information from the eye to the brain. Horizontal cells contact both the receptor cells and the bipolar cells, so they make a horizontal connection. Amacrine cells contact the bipolar cells and ganglion cells for an inhibitory interaction within the retina. Both of these are specialized in lateral interactions within the retina instead of straight back, so they're like this. All cells, so far except for ganglion cells, generate graded local potentials. Neurons affect each other with these graded release of neurotransmitters in response to graded changes in electrical potentials. In opposition to all the cells that were discussed so far, ganglion cells generate action potentials. There's two different types of systems. There's the scotopic system, which operates best at low levels of light and mostly involves the rods. Then there's the photopic system, which mostly involves the cone, and it is sensitive to color. Next section is photoreceptors transduce light into chemical reactions. Sensitivity of rods and cones is due to their biochemistry. They look like stacked discs. Fractions of light that pass through there's only a fraction of light that passes through the cornea that reaches the retina. The stacked disc structure increases the likelihood that one will capture those light particles that reach the retina. When the photons strike the disc, they're captured by special photopigment receptor molecules. One of these is rhodopsin. So when light hits rhodopsin, retinal dissociates from opsin, which are the two components of rhodopsin, to reveal an enzymatic site. The altered opsin then activates molecules of G protein transducin. Transducin then acts through enzyme phosphodiesterase, or PDE, to break apart cyclic GMP to 5' GMP. Cyclic GMP then holds cyclic GMP in its inherent form holds channels for sodium open. The stimulation by light then closes these channels and creates a hyperpolarizing event. When the light hitting rhodopsin via this pathway hyperpolarizes the it hyperpolarizes the rods. The size of the hyperpolarizing photoreceptor potential determines how much less synaptic transmitter will be released. And then those two subcomponents of rhodopsin are opsin and retinal. Visual receptor molecules span the membrane of the discs and have structures that are similar to G protein coupled neurotransmitter receptors. Due to this whole system, compromises the three characteristics of the visual system. The first being sensitivity to weak stimuli, in which weak stimuli are amplified to produce physiological effects. The second being the integration of stimuli over time, which makes vision relatively slow but increases its sensitivity. And the last being adaptation to a wide range of light sensitivities. The next section discusses just the basics of light. The physical energy our visual system responds to is just a band of electromagnetic radiation, which usually spans 400 to 700 nanometers that we can respond to. Quanta are a discrete unit of electromagnetic energy. Each can be described by a single number. Each quanta can be described by a single number. And the wavelength is the distance between the two peaks in a repeated stimulus. Photons are quantum of electromagnetic energy in the range of wavelengths we call light. Each has a tiny amount of energy and the exact amount of energy depends on the wavelength. The next section is different mechanisms enable the eye to work over a wide range of light intensities. The visual system in general deals with a wide range of light intensities. The first way that it deals with this is to adjust the size of the pupil. For example, when bright light hits the pupil, it constricts. This is called a pupillary response, which is controlled by retinal ganglion cells, which on their own possess their own photopigments. These retinal ganglion cells project to the superior colliculus to control pupil diameter and match the level of ambient light. The next way is through range fractionation, which we discussed earlier, which is a lot of cells working in parallel to respond to different intensities of light so that there's a lot working at the same time. The last being adaptation, which is a progressive loss of receptor sensitivity as stimulus is maintained. Each photoreceptor adjusts its sensitivity to match the ambient illumination, similar to the pupillary response. Photoreceptor adaptation specifically is the tendency of rods and cones to adjust their light sensitivity to match ambient levels of illumination. This is the main reason we can see over wide ranges of light. There's two main factors that account for the adaptation of the photoreceptor. First being varying concentration of calcium ions to control sensitivity to light. And the second being photoreceptor pigment is split apart by light, and then its two components, which are retinal and opsin, recombine slowly, which affects how much photopigment is available to respond to light. Last section is acuity is best in the foveal vision. The visual field is overall the whole area that you can see without moving your head and eyes. Visual acuity is the sharpness of vision. The fovea is a central portion of the retina with a dense concentration of cones, and it's the center of our gaze. It has a very high visual acuity there. People differ in their concentration of cones, so we all have differences in visual acuity. Noctic disc is the region of the retina devoid of receptor cells because ganglion cells, axons, and blood vessels exit the eyeball here. This is called the blind spot because we can't see anything there. Saccades are fast movements of your eye that present various parts of the visual scene to the fovea. When we fix our gaze, there's still small saccades that we are unaware of that avert photoreceptor adaptation. If the saccades didn't continue to happen, if we stared at something for a long time, we'd eventually not be seeing anything because there would be an adaptation of the stimulus. Rods aren't in the fovea at all. They're only present in the periphery. So we're more sensitive to dim light, and it increases our system sensitivity to weak stimuli. And that is Chapter 10, Segment 1.